Hello friends, welcome to the world of Ignite Brain co-powered by Need Capsule. Today we are discussing the chapter that is Biomolecules, one of the best video for biomolecules in one shot. Let's start. Biomolecules, living organism, chemical composition. सेल को ग्राइंड करेंगे ट्राईक्लोरो एसेटिक एसिड, इसका मॉलिकुलर फार्मूला होगा Cl3CCCOOH। ट्राईक्लोरो एसेटिक एसिड को फिल्टर करेंगे, एक आएगा जो फिल्टर हो जाएगा एसिड सॉल्युबल पूल होगा और जो फिल्टर नहीं होगा एसिड इनसॉल्युबल पूल होगा। बायोमोलिकुल्स दो पार्ट्स में डिवाइडेड इज माइक्रोमोलिकुल और मैक्रोमोलिकुल्स। Micromolecules present in acid soluble pool और macromolecules present in acid insoluble pool। अगर माइक्रोमोलिकुल्स लेस देन 800 डाल्टन मॉलिकुलर वेट से कम होगा तो माइक्रोमोलिकुल्स के अंदर आएंगे और मैक्रोमोलिकुल्स के अंदर आएंगे जो 1000 डाल्टन से ऊपर होगा। एग्जाम्पल में आ जाएगा माइक्रोमोलिकुल्स के अंदर माइक्रोमोलिकुल्स का एग्जाम्पल हो जाएगा अमीनो एसिड, नाइट्रोजन बेस, मोनोसेक्राइड्स। और मैक्रोमोलिकुल्स का एग्जाम्पल हो जाएगा प्रोटीन, न्यूक्लिक एसिड और पॉलीसेक्राइड्स। बहुत ही इंपॉर्टेंट पॉइंट्स यहाँ लिखा हुआ है। लिपिड्स आर माइक्रोमोलिकुल्स बट दे आर फाउंड इन एसिड इनसॉल्युबल पूल। लिपिड्स होता है 1000 डाल्टन से ऊपर होता है, फिर भी माइक्रोमोलिकुल्स के अंदर रखा हुआ है, बट दे आर फाउंड इन एसिड इनसॉल्युबल पूल। अब नेक्स्ट पॉइंट क्या है, दिस was for carbon compound found in living molecules। इनऑर्गेनिक कंपाउंड बर्न होकर कार्बन बनाएगा, कार्बन ऑक्सीडाइज होकर CO2 में कन्वर्ट होगा और जो रिमेनिंग पार्ट्स रह जाएगा एस जो इनऑर्गेनिक कंपाउंड है। सम इनऑर्गेनिक कंपाउंड इज ऑल्सो फाउंड इन एसिड soluble pool, that is sulfate and phosphate। Most of the question asked from this line, most abundant inorganic compound in earth crust that is silicon 27.7% और एटमॉस्फेयर में कौन है ऑक्सीजन that is 46.3%। in human body oxygen is 65% by weight। अब हम लोग समझने वाला है प्राइमरी एंड सेकेंडरी मेटाबोलाइट्स के बारे में। प्राइमरी और सेकेंडरी मेटाबोलाइट्स कौन कौन सा है। प्राइमरी मेटाबोलाइट्स आर सबस्ट्रेट दैट इज रिक्वायर्ड फॉर प्राइमरी मेटाबॉलिक एक्शन। प्राइमरी मेटाबोलाइट्स का एग्जाम्पल कौन कौन सा है, प्रोटीन, लिपिड, कार्बोहाइड्रेट। सेकेंडरी मेटाबोलाइट्स हैव नो डायरेक्ट सिग्निफिकेंस इन प्राइमरी मेटाबॉलिक एक्शन यूटिलाइज बाय अस फॉर इकोनॉमिक पर्पस। Some secondary metabolites are pigments। पिगमेंट्स में कौन कौन सा आ जाएगा, केराटिनॉइड्स और एंथोसेनिन, डेट इज केराटिनॉइड्स फॉर रेड, एंथोसेनिन फॉर ब्लू। एल्कोलाइड्स में हो जाएगा मॉर्फिन और कोडीन। टर्पेनॉइड्स में हो जाएगा मोनोटर्पिन्स, डाइटर्पिन्स। और इसेंशियल ऑयल में हो जाएगा लेमन ग्रास ऑयल। टॉक्सिन में हो जाएगा एडविन और रिसिन। लेसेथिन मोस्ट कॉमनली आस्क्ड दैट इज कोनाकाविलिन ए। ड्रग्स में हो जाएगा विनब्लास्टिन और क्रुक्रूमिन। पॉलीमरेज में हो जाएगा रबर, गम्स, सेलुलोज। अब जो नेक्स्ट टॉपिक है प्रोटीन के ऊपर है। प्रोटीन मेड अप ऑफ CHON कौन, इसेंशियल एलिमेंट्स इन प्रोटीन। Protein made up of con. Trick is CHON, con. Essential elements in protein. Most abundant protein in biosphere, that is Robisco. Most abundant protein in biosphere, that is Robisco. Most abundant protein in animal world, that is collagen. Protein are heteropolymers of amino acid. Structure of amino acid. Substituted, another name of amino acid is substituted methane. Structure of amino acid is very important. NH2 group, that is amine group, and COOH, that is carboxylic group, and R is functional group. One example is also given there. Example of disaccharide co-powered by neat capsule. We can learn the example of disaccharide with the help of trick, that is mala D steroid tablet. M stand for maltose and L stand for lactose. D stand for disaccharide. D stand for disaccharide that identified the example of disaccharide mala D. Ma stand for maltose and La stand for lactose, and steroid stand for sucrose. T stand for trehalose. Simplest amino acid, that is glycine, and H group identify that example of glycine, and NH2 group, COOH group, and hydroxyl group identify that example of alanine. NH2 and COOH and CH2OH group identified the structure of serine. Total 20 types of amino acids are found, 10 essential and 10 non-essential amino acids. Two amino acid, that is semi-essential amino acid, that is arginine or histidine. Essential amino acid must be present in our diet, that is methionine, threonine, valine, phenylalanine, isoleucine, tryptophan, lysine, leucine, and histidine. We can learn example of essential acid with the help of trick. Trick is co-powered by neat capsule, that is PBT, team, Hall. P stand for phenylalanine, V stand for valine, or T stand for threonine, or T again stand for tryptophan, and I stand for isoleucine, and M stand for methionine, and H stand for histidine, and A stand not given there, and L stand for leucine, and again L stand for lysine. Non-essential amino acid, that is glycine, serine, alanine, cysteine, aspartic acid, glutamic acid, tyrosine, proline. इसके बाद हो जाएगा तुम्हारा asparagine and glutamine. Classification of amino acid, that is acidic amino acids are glutamic acid and aspartic acid. They have one amino group and two carboxylic group, NH2, COOH, and COOH. एरोमेटिक अमाइनो एसिड का एग्जाम्पल हम लोग ट्रिक से कैसे याद करें. ट्रिक इज को पावर्ड बाय नीट कैप्सूल, दैट इज पार्टी PATTY. P stand for phenylalanine, A stand for aromatic, T stand for tryptophan, and TY stand for tyrosine. Deuter ion, both positive and negative charge. NH3 positive and COO negative. Basic amino acids, two amino group and one carboxylic group, that is NH2 and COOH. Example of basic amino acids are arginine, histidine and lysine. बेसिक अमाइनो एसिड का एग्जाम्पल है arginine, histidine and lysine. Histidine used for histone protein in DNA packaging. Rest is amino acid are neutral amino acid. Rest amino acids are neutral amino acid, that is valine. Most complex amino acids that are tryptophan, derivative of melatonin and serotonin. Tyrosine क्या है? मेलानिन पिगमेंट, थायरोक्सिन और एड्रेनालिन और नॉन एड्रेनालिन. Aromatic amino acids are tyrosine, tryptophan, phenylalanine have benzene in their functional group. Example of non-protein amino acid. Trick is co-powered by neat capsule, that is Congo, CONGO, Congo. C stand for citrulline, O stand for orthinin, and N stand for non-protein amino acid, and G stand for GABA. Proteins are heteropolymers made up of different types of amino acid. Protein are heteropolymers made up of different types of amino acid. This can be peptide bond, COON, C single bond N. Configuration of protein molecule. Configuration of protein molecules. Primary configuration: a straight chain of amino acid linked by peptide bond only. Primary configuration: a straight chain of amino acid linked by peptide bond only. Example: newly formed protein, structurally unstable. Secondary configuration: peptide bond plus hydrogen bond. Example of homopolysaccharide co-powered by neat capsule. Example with the help of trick, that is DISC, CG. D stands for dextrin, I stands for inulin, S stands for starch, and C stands for cellulose, and G stands for glycogen, and C stands again for chitin. Alpha helix, right-handed spirally rotated structure formed due to intermolecular hydrogen bonding. Alpha helix, right-handed spirally rotated structure formed due to intermolecular hydrogen bond, that is between the amino acid of the same chain. Example of alpha helix, that is myosin, tropomyosin and keratin. Example of alpha helix, that is myosin, tropomyosin and keratin. Second one is beta helix or beta plated, that is zigzag structure like this, formed by intermolecular hydrogen bonding. Example is silk, that is fiber and keratin. Tertiary structure, tertiary structure peptide bond plus hydrogen bond plus disulfide bond. Tertiary structure peptide bond plus hydrogen bond plus disulfide bond. Sulfur containing amino acids are methionine or cysteine. Sulfur containing amino acids are methionine or cysteine necessary for disulfide bond. All enzymes are tertiary protein and they show a globular appearance. All enzymes are tertiary protein and they show a globular appearance. Example of globular proteins. We can learn the example of globular proteins with the help of trick co-powered by neat capsule, that is Garmi. Garmi trick is Garmi. GHARMII. Garmi. G stand for globulin, H stand for histone, A stand for albumin, R stand for Robisco, M stand for myosin, and I stand for inulin. Quaternary structures. Two or more polypeptide chains of tertiary structure are joined. Quaternary structure. Two or more polypeptide chains of tertiary structures are joined. That is example of quaternary structure, that is insulin 51 amino acid, glucagon 29 amino acids. Note, quaternary structure is the most stable structure because of the maximum number of bond. Quaternary structure is the most stable structure because of maximum number of bond. Peptide chain N-terminal and C-terminal. First for N-terminal for amino acid and C last carboxylic group. Types of protein. Types of protein: simple, compound और derived. Types of protein: simple, compound, derived. फाइब्रस हो जाएगा कोलेजन, इलास्टिन और केराटिन. कोलेजन हो जाएगा कनेक्टिव टिश्यू जो टेंडन का पार्ट होता है, इलास्टिन जो लिगामेंट और केराटिन स्किन में पाया जाता है. ग्लोबुलर प्रोटीन रोबिस्को, एल्ब्यूमिन, ग्लोबुलिन और हिस्टोन. Histone help in DNA packing. Globulin present in hemoglobin and albumin blood colloidal osmotic pressure को मेंटेन करता है albumin. Vials have lower melting points, that is Gingelli vial. Two nucleotides are joined by 3-5 by phosphodiester linkage. Compound protein part made up of amino acid और नॉन प्रोटीन पार्ट. प्रोटीन पार्ट का एग्जांपल हो जाएगा तुम्हारा न्यूक्लियोप्रोटीन, क्रोमोसोम डीएनए प्लस आरएनए प्लस प्रोटीन, राइबोसोम आरआरएनए प्लस प्रोटीन. GLUT4. Most of the question asked from this line. GLUT4 helps in glucose transport in a cell. GLUT4 help in glucose transport in a cell. Next topic is nucleic acid. Polymers of nucleotide. Nucleic acid is the polymer of nucleotides. नाइट्रोजन बेस प्लस पेंटोज शुगर प्लस फॉस्फेट ग्रुप मिलकर बनाएगा न्यूक्लियोटाइड, एंड नाइट्रोजन बेस प्लस पेंटोज शुगर बनकर बनाएगा न्यूक्लियोसाइड. नाइट्रोजनस बेस आर प्यूरिन और पायरिमिडीन. प्यूरिन क्या होता है? 9 member डबल रिंग स्ट्रक्चर. प्यूरिन इज 9 member डबल रिंग स्ट्रक्चर. नाइट्रोजन इज फाउंड एट position of 1379. Nitrogen is found at 1379. Trick for remember the purine is AG, that is adenine or guanine. Pyrimidine, that is 6-member single ring structure. 6-member single ring structure. Nitrogen is at the position of 1 or 3. Nitrogen is present at the position of 1 and 3. Example of pyrimidine is cut, cut, that is cytosine, uracil or thiamine. Example of pyrimidine is cut, that is cytosine, uracil or thiamine. Bond between phosphate and hydroxyl group is ester bond. Bond between phosphate and hydroxyl group, that is ester bond. Example of pyrimidine, that is cysteine, uracil or thymine. Cytosine, uracil and thymine. This is the structure of cytosine, and this is the structure of uracil, and this is the structure of thymine. Example of purine, that is adenine. This is the structure of adenine, and this is the structure of guanine. Pentose sugar, deoxyribose, that is DNA. H at second position, deoxyribose, and OH at second position in RNA, that is ribose sugar. Adenine and guanine are substituted purines. Phosphate group. Nitrogen base plus pentose sugar, that is nucleoside. Nitrogen base plus pentose sugar, that is nucleoside. Nitrogen base plus pentose sugar are nucleoside. Nitrogen base plus pentose sugar plus phosphate group, that is nucleotide. The common mistake between nucleoside and nucleotide, that is nitrogen base plus pentose sugar, that is nucleoside, and nitrogen base plus pentose sugar plus phosphate group, that is nucleotide. Structure of nucleotide. In glycosidic linkage between nucleotide, 5 phosphoester linkage. 2 nucleotides are joined by phosphodiester linkage. Two nucleotides are joined by phosphodiester linkage. Trick for disaccharides are so simple. Trick is co-powered by neat capsule. This is clear visible to everyone. Yeah. Trick for disaccharides are, that is lac suma tree. LA के लैक्स हो जाए लैक्टोज, SU से हो जाएगा सुक्रोज, MA से हो जाएगा माल्टोज, एंड ट्री से हो जाएगा ट्राइहेलोज. First amino acid is called N-terminal. Last amino acid is called C-terminal. Lipid term given by Bloor, CHO, but H or O का ratio not equal to 2:1. CHO but H or O का ratio not equal to 2:1. Lipid do not form polymers. Lipids are not strictly macromolecules. Lipids are not strictly macromolecules. Simple lipids or neutral fat, that is fatty acid, glycerol. These are ester of long fatty acid and alcohol. Alcohol, that is glycerol. These are ester of long chain, long fatty acid and alcohol, that is glycerol. One fatty acid plus one glycerol, that is monoglycerides. Two fatty acid plus one glycerol, that is diglycerides. Three fatty acid plus one glycerol, that is triglycerides. Another name of glycerol, that is trihydroxypropane. Another name of glycerol, that is trihydroxypropane. Simple lipid contain two types of fatty acid. Simple lipid contain two types of fatty acid. First one is saturated fatty acid that have single bond. Saturated fatty acid have single bond. Only single bonds are present between C atom. C single bond, C atoms. That is कार्बन एटम के बीच में सिंगल बॉन्ड रहेगा. नॉन इसेंशियल फैटी एसिड, नॉन इसेंशियल फैटी एसिड, मेटाबॉलिकली लेस एक्टिव, palmitic acid, that is 16 carbon, palmitic acid, that is 16 carbon, and stearic acid, that is 18 carbon. Palmitic acid, that is 16 carbon, and stearic acid, that is 18 carbon. Unsaturated fatty acid, trick is ओला उबर. अनसेचुरेटेड फैटी एसिड का एग्जांपल है ओला उबर. ट्रिक इज को पावर्ड बाय नीट कैप्सूल. ओला, OLAUBER, उबर, ओला उबर. ओ के ओ से स्टैंड करेगा ओलिक एसिड, एल से हो जाएगा लियोनेलिक एसिड, एंड ए से हो जाएगा आर्किडोनिक एसिड, एंड उबर के हो जाएगा अनसेचुरेटेड. उबर आइडेंटिफाइड द ट्रिक ऑफ अनसेचुरेटेड फैटी एसिड. डबल बॉन्ड कार्बन एटम इसेंशियल होता है. ये मेटाबॉलिकली एक्टिव होता है. ओलिक एसिड में हो जाएगा सिंगल डबल बॉन्ड. लायोलेनिक में हो जाएगा 2, लियोनिक एसिड में हो जाएगा 3, और आर्किडोनिक एसिड में हो जाएगा 4, जो 20 कार्बन का होता है. नंबर ऑफ डबल बॉन्ड्स can be asked in exam, that is oleic acid 1, limonenic acid, that is 2, and arachidonic acid, that is 4. Conjugated lipid, that is phospholipid, that is lecithin. Phospholipid, that is most abundant type of lipid in protoplasm. Phospholipid, most abundant type of lipid in protoplasm. Two molecules of fatty acid plus glycerol plus H3PO4 plus nitrogenous compound. Derived of lipid, that is steroids, that is tetracycline structure. Estrol, that is cholesterol. मोस्ट डेकोरेटेड माइक्रोमोलिकुल्स. कोलेस्ट्रॉल क्या है? मोस्ट डेकोरेटेड माइक्रोमोलिकुल्स. कोलेस्ट्रॉल हेल्प्स इन सिंथेसिस ऑफ विटामिन डी. कोलेस्ट्रॉल हेल्प्स इन सिंथेसिस ऑफ विटामिन डी. नाउ another topic is carbohydrates. CHO. HO का रेशियो 2:1 होगा. that is hydrates of carbon. Carbohydrates are sugar. Monosaccharides, simplest sugar that can't be further hydrolyzed. Free aldehyde group, that is aldose. Free keto group, that is ketose. Example of monosaccharides are DHAP, that is arthulose and xylose. All monosaccharides are reducing sugars, that gives Vendic taste or Failing taste for for testing of glucose in urine. जो टॉलिंस रिएजेंट्स और फैलिंग सोल्यूशन को जो रिड्यूस कर जाएंगे उसको क्या बोलते हैं? फैलिंग सोल्यूशन और टॉलिंस रिएजेंट्स को जो रिड्यूस कर जाएंगे उसको क्या बोलते हैं? रिड्यूसिंग शुगर. Triose is 3 carbon compound, that is example of triose is DHAP and PGL. टेट्रोज का एग्जाम्पल हो जाएगा अर्थेोज. पेंटोज का हो जाएगा राइबोज और डीएक्सीराइबोज. हेक्सोज का एग्जाम्पल हो जाएगा ग्लूकोज, फ्रुक्टोज और गैलेक्टोज फाउंड इन ब्रेन शुगर. स्वीटेस्ट शुगर इज फ्रुक्टोज. हेप्टोज लार्जेस्ट मोनोसैकेराइड्स, that is pseudohaptose. Oligosaccharides made up of 2 or 10 monosaccharides units. Oligosaccharides made up of 2 to 10 monosaccharides unit joined by glycosidic linkage. Oligosaccharides are joined by glycosidic linkage. Disaccharides, that is maltose, glucose plus glucose joined by 1,4 glycosidic bond. Sucrose, that is invert sugar, glucose plus fructose, that is non-reducing. Lactose, glucose plus galactose. Lactose क्या है? Glucose plus galactose. Lactose is glucose plus galactose. Starch gives blue color in presence of iodine because of its helical structure which can store I2. Starch gives blue color in presence of iodine because of its helical structure which can store I2. Next our topic is polysaccharides. Homopolysaccharides. Cellulose क्या है? Homopolymer. Cellulose is homopolymer, linear polymer of beta D glucose. And cellulose is homopolymer. Most abundant organic material is cellulose. Most abundant organic material is cellulose. Starch stored food in plants and animals in glycogen. Amylose 20 to 250 to 30 unbranched, and amylopectin, that is branch, that is 330 glucose unit. Amylose gives blue color to iodine. Amylopectin, that is red colored iodine. Starch from helical structure, so they can hold I2, but cellulose can't hold iodine. Glycogen, animal starch. Glycogen क्या है? Animal starch give red color with iodine. Glycogen animal starch give red color with iodine. Chitin, homopolysaccharides. Chitin क्या है? Homopolysaccharides made up of N-acetyl glucosamine. Chitin homopolysaccharides made up of N-acetyl glucosamine forms the exoskeleton of arthropods. That is jointed appendages. Right end in polysaccharide is reducing end. Right end in polysaccharide is reducing end, and left end in polysaccharide are non-reducing end. Inulin क्या है? Homopolymer. Inulin is homopolymer, linear polymer of fructose. Inulin क्या है? Linear polymer of fructose. Water soluble polysaccharides. Inulin is linear polymer of fructose. Water soluble polysaccharide. Inulin is homopolymer. Next is heteropolysaccharide. Next is hydropolysaccharide. Our trick is 4 HP computer. Trick is co-powered by neat capsule 4 HP computer. 4 HP मतलब 4 times H repeated, 1 2 3 4. H hydropolysaccharides, H hemicellulose, and H again heparin, and H is hyaluronic acid, and C stand for C, and P stand for packaging. Example is hyaluronic acid, heparin, packaging, and hemicellulose. Mucopolysaccharides, that is peptidoglycan in bacterial cell wall, N-acetyl glucosamine and N-acetyl muramic acid. Now our topic is enzyme. Enzymes, tertiary protein. Due to this structure, it has active sites or pockets. Tertiary protein. Due to this structure, it has active sites or pocket. First enzyme discovered by Gymase. First enzyme discovered was Gymase. Active site. This is the active site. Active sites where substrate B binds. Where active site where the substrates binds. Quaternary structures, active sites present, they are isoenzyme. Quaternary structure, active sites present, they represent the isoenzyme. Cellulose does not contain complex helix and cannot hold I2. Cellulose does not contain complex helix and cannot hold I2. Enzymes are very specific in nature to their substrate. The catalytic property of enzymes depends on two factors. The catalytic property of enzymes depends on two factors. Turnover number. It is the number of substrate molecule convert into product per unit. Turnover number, it is the number of substrate molecule convert into product per unit time. Carbonic anhydrase is the fastest enzyme. Carbonic anhydrase is the fastest enzyme. Km constant, that is Michel's constant. This substrate concentration where the velocity of enzymatic action will be Vmax. The graph of Km constant, that is velocity of reactions and substrate concentration. Km Vmax, velocity of enzymatic action maximum. जब Km का वैल्यू इंक्रीज करेगा तो enzymatic एक्टिविटी घटेगा. जब Km वैल्यू इंक्रीज करेगा तो enzymatic एक्टिविटी डिक्रीज करेगा. Removal of CO2 from amino acid, making an amino acid into amine. Removal of CO2 from amino acid, making an amino acid into amine. Catalytic power of an enzyme is inversely proportional to Km value. Catalytic power of an enzyme is inversely proportional to Km value. How do enzymes bring about such high rate of chemical conservation? One substrate break 10 joule energy, enzyme break 2 joule energy. This is the activation energy is without enzyme and activation energy without enzyme. That is the energy and progression of reaction, that is product. Holoenzyme equal to apoenzyme plus cofactor. Holoenzyme equal to apoenzyme plus cofactor. Nature of enzyme action: substrate plus enzyme. Nature of enzyme action: substrate plus enzyme. That is, can be enzyme substrate complex and enzyme product complex. After that, enzyme plus product. To decrease the activation energy of reaction, cofactor, नॉन प्रोटीन पार्ट. कोफैक्टर क्या होता है? नॉन प्रोटीन पार्ट. एंजाइम, सिंपल एंजाइम एंड कंजुगेटेड एंजाइम. Enzyme, simple enzyme made up of protein only. Conjugated enzyme, holoenzyme. Conjugated divide into two types: apoenzyme and cofactor. Apoenzyme is protein part, cofactor is non-protein part. Cofactor further divided into three types, that is coenzyme, prosthetic group, or metal activates. Coenzyme, organic compound loosely bound to apoenzyme. Its association with apoenzyme is transient in nature. Prosthetic group, organic compound tightly bound to apoenzyme. Prosthetic group, organic compound tightly bound to apoenzyme. Metal activates, inorganic. Coenzyme, most of them are vitamin derivatives. Prosthetic group, peroxidase catalase, that is prosthetic group, that is heme. Metal activators, Zn2 plus, that is alcohol dehydrogenase, carboxypeptidase, and carbonic anhydrase. Molybdenums, nitrogenase. And classification and nomenclature. Oxidoreductase and dehydrogenase, enzyme which are oxidoreductase, that is classification and nomenclature. Oxidoreductase, enzyme which are oxidoreductase reacts S reduce plus S dash oxidase gives S oxidase plus S dash reduced. S is substrate. Example is dehydrogenase. अगर वीडियो अच्छा लगा हो तो वीडियो को लाइक करें, और चैनल पर नए हैं तो चैनल को लाल बटन दबाकर चैनल को सब्सक्राइब करें और दोस्तों में शेयर करें. जब यह वीडियो रिकॉर्ड हो रहा है, बज रहे हैं अभी रात के 12:30.